Hi, Subhoja. Thanks for joining me today.
Of course, yeah, my pleasure. Thank you so much for giving me the opportunity to share my research and so that we can have a very enthralling discussion in order to guide students who would be enthusiastic about this.
Definitely. Yeah, this podcast is geared towards undergraduate chemists and researchers in other scientific fields, and I
Mhm.
think they'll find your work to be very interesting. To start with, could you give us a summary of your current research focus and its significance in the fight against malaria?
Of course. So, malarial drug discovery is one of the projects that I'm currently a part of at the University of Central Florida since I began my postdoctoral tenure here 2 years ago. Before that, I did my PhD in structure based drug designing in malaria, which mainly involved identifying different targets on which work had already been done, but seeing them in a structural light so that we are able to target them better using in silico methods, recombinant protein expression, X ray crystallography to get the exact crystallized 3D structure of the target and the ligand or the inhibitor that we are planning to target it with. So that gave me a more structural perspective of how we could approach drug designing. Now I'm more into drug discovery that involves working with different compounds. And when I say compounds, I definitely mean from a perspective of repurposing them. The latest publication from my current lab that we had about a month ago mainly focuses on a repurposed mammalian CDK like kinase inhibitor. So, we are already familiar with different kinase inhibitors having been used in other diseases like cancer or even cardiovascular stuff. But those being reimagined as inhibitors against malarial drug targets is something very novel that I came across in this lab. So our team of collaborators, they keep synthesizing these compounds so that they can be made better and more specific in targeting their drug targets of interest in the malarial milieu. And eventually, by tailoring those compounds in that way, we can use them to be more specifically targeted towards those particular kinases. And when you ask about why kinases, it comes across that kinases are ubiquitously present across different pathways, especially in cell signaling and in several growth pathways. They have multiple downstream phosphorylating targets, and so that targeting a particular pathway or a particular kinase in that pathway gives us an advantage that we can achieve in any one of the particular life cycles of malaria. Malarial life cycle is also quite complex when we talk about that. It has a primary host, which is the mosquito. It completes a set of stages there. Then it comes to the secondary host, which are the humans, in which it completes its life cycle in both its liver cells or hepatocytes and also the RBCs or erythrocytes. They have their different, sexual and asexual stages sequestered between all of those hosts. And targeting a particular pathway could be gametocyte differentiation, could be related to gametocytogenesis, could be related to an asexual stage. If it gives us an advantage that the growth of the parasite can be regulated, can be achieved like maybe over 30-40% of growth inhibition can be achieved, then that compound can be used for other in vitro assays, in vivo assays to give us more details into how specific that particular compound is. So, as I've come to know through my time in this lab, that this lab has particularly invested in different kinds of kinase inhibitors, be it limb kinase, targeted against limb kinases, kinase inhibitors, and all of these, which have already been implicated in other diseases. But as I said, they can be reimagined and repurposed as antimalarials as well. So that's in a nutshell.
In the three pieces of research I reviewed, it was clear to me that chromatography played a critical role in these studies. How did the related choices impact the quality and the yield of your target complexes?
So when I talk about chromatography, I have dabbled in chromatography since my master's days while I was a grad student, as I have visualized how to do simple stuff like paper chromatography. That has really intrigued me into how it has spread its wings across identifying and separating different biomolecules as well. As I got to experience affinity chromatography, that was one of my first trysts with proper research based chromatography work, while I was trying to purify recombinant proteins. using nickel resin, using cobalt resin. It really got me intrigued that yes, you can have very pure proteins extracted and purified, and you can work with it in a very efficient manner. Eventually, I got into FPLC, where I worked with pre-packed columns and a very compact way of getting through all the stepwise procedures in an assembly line for different recombinantly expressed proteins. For the different in vitro assays, we do try to have different recombinantly expressed proteins expressed and purified through chromatography. So a bit of affinity chromatography, a bit of gel filtration or size exclusion chromatography, ion exchange, and all of that. That helps us get as pure protein as we can get. But one bottleneck that I have definitely experienced is expressing high molecular weight proteins. So the chances of it being expressed more in the inclusion bodies or getting more into the insoluble fractions, that keeps happening. So we need to have a very careful choice of the vectors of the expression systems so that we can get as high yielding or high yield of a protein as we can for our particular protein of interest. So yeah, chromatography definitely, for routine lab work, it forms a part of my lab work as well for expressing different kinds of proteins and getting them purified for in vitro assays. It definitely is a very important tool in my research work.
And that's a great transition. I don't know a whole lot about size exclusion chromatography, so I was hoping you could talk about how you integrate that into your workflow.
The main problem is not the chromatography, but the expression of your proteins, right? How much protein you're being able to express with the right IPTG concentration and the correct vector and the correct expression system that will help you get your protein in bulk amounts. Previously, when I used to work with crystallography, I needed to have a large amount of pure protein. And for that, I had to pass my lysate through the nickel beads to get affinity chromatography purified. After getting all those fractions, it would still be contaminated with multiple lower molecular weight bands. So the bottom line is, as you keep purifying proteins, as you keep expressing them, you have to keep checking them on a gel to see how pure your lysate is or how much of your protein of interest has it got. And then you try to eliminate all the different outliers to get your particular band of interest in as pure as it can be, because there are chances of contamination as well. It can arrive. You know what happened once? I had contamination in my sample buffer while I was trying to run my protein on a gel. And due to contamination in my sample buffer, I was getting multiple bans on my SDS page. And I was like, what's happening to my protein? It's like a not more than a 30 kilodalton protein. And I'm getting all these different bans. So you keep troubleshooting, right? All of this is a part of your learning curve. All of this is a part of your experience, and it goes a long way the more you troubleshoot it yourself. So getting back to what I kept doing, size exclusion, as I said, two of the main techniques that I followed for size exclusion was after getting the protein initially purified through affinity chromatography, I had to desalt them because usually for the elution step of an affinity chromatography, you need to have a high concentration of, if it's a GST tagged protein, then you need to have glutathione in your elution buffer. Or if it's a nickel a his tagged protein, then you need to have imidazole in your buffer. So you need to get rid of that imidazole or glutathione so that it doesn't interfere with the activity of your protein. In order for that to do, used to purify the eluted fractions through maybe a G25 column or a PD10 column to get them desalted so that all the salt would pass off and I would be left with more of my protein. But in order to get rid of other contaminating bands, it was also important that I ran through even finer beads like a Superdex 30 by 100 or a Cephacryl S100 column to get rid of those lower contaminating bands and get my eventual protein of interest that I need. And it did not stop there. In order to have more concentrated proteins, I needed to concentrate my protein as well, using the perfect molecular weight cutoff that I needed. So if you're trying to purify or concentrate a 30 kilodalton protein, then you need to have a molecular cutoff like 10 kilodalton. Or if it's a higher molecular weight protein, then you need to have something around like 30 kilodalton. So you keep building up, you keep troubleshooting, you keep dabbling with all these different tools. And I think I must appreciate the way biotechnology has stepped up our game at every step of the way. The more interdisciplinary it has become, all the tools that we get at our fingertips, and we can actually achieve all these techniques done, we get our results like in no time. So remember reading about how Carey Mullis got the idea of PCR while he was traveling down some beach in California. And in those days, there was no like thermal cyclers available. So they had to use all these water baths to get through all these temperatures. And it used to be such a cumbersome work. And these days, you have things like digital PCR, where, even differences in like one CT value gives you like a huge change in expression or your signal amplification. And it's fascinating. It's wonderful. I'm just so glad to be a part of this entire world of biotech and how things are amping up and how healthcare is going great guns, I believe. So,
Mass spectrometry is mentioned as a tool that's helpful for your sample characterization.
mhm.
Could you elaborate on how MS fits into your research pipeline?
Yes. I could cite one example since I'm already involved in cancer research as well. So that also involves identifying different targets. So anything that you start from, maybe chromatography, maybe mass spectrometry, immunoprecipitation, or some other pull down experiment, which are the core items of interdisciplinary biological research these days. You have to start with the whole cell lysate, right? And you have to boil down to all those targets. Mass spectrometry in that case can definitely help us in understanding whether biomolecular recognition, biomolecular target validation can be achieved that way. So, once we are working with whole cell lysates, we need to have different affinity tags bound to different targets. Even cell lines can be made in that way that your protein of interest or your target of interest can maybe have an HA tag, maybe through a dicree PLOX system expressed that way. There could be different ways you can use, maybe like native mass spectrometry. You can also use affinity based mass spectrometry. And these days, I think mass spectrometry also helps us in understanding different interactors. So, identifying those, maybe you can use a CRISPR Cas9 tech. But eventually, mass spectrometry will definitely help you in understanding what exactly it binds to. Suppose I'm working with long non-coding RNAs, okay, and I want to find out that expressing this particular long non-coding RNA can help us understand which RNA binding proteins it might interact with. In that case, using an antibody based pull down and then using the pull down product in mass spectrometry to find out the enrichment of certain proteins can definitely help us in understanding what are the interactors of that particular non coding RNA is. So, it spreads across a vast array of biomolecules, a vast array of pathways. Even pathways can definitely help in that way. So yeah, I think these days LCMS, high throughput sequencing, then using them for mass spectrometry. And also, I would like to mention another technique that we have come across in recent times, like affinity based probing, turbo ID generation, then proximity labeling. These also involve mass spectrometry to a great detail, but everything is kind of directing towards finding out the exact interactors and then validating them using different, you can maybe use a knockout model, you can use an express model, and it definitely helps that way.
Makes sense. Now, I know this is a pretty broad question, but I'm always curious to hear about what sample preparation challenges you have to overcome. Certainly, you're working with forming stable protein complexes. There's a lot that can go wrong, I suppose. So I was curious about that.
Undoubtedly. So the bottom line of any biological experiment is to have at least three bioreplicates, right? So it might go right one time and it might go wrong the other two times. But that doesn't mean that you have not got the results from there. So, yes, building something from hypothesis to validation definitely takes time. And when you're working with biological samples, they are pretty rough to begin with. So, I know these days we are doing cell culture, we can harvest cells very well, then it might take you like an entire day to prep a sample. So while working with whole cell lysates, it might contain different, DNases, RNases, proteases that might tamper with your proteins, and your protein might get, degraded very fast. So in order to prevent that degradation, we have to be extra careful in handling them, and at the same time, cutting down the time short so that it doesn't take longer, or maybe your protein or your exact sample that you're preparing isn't at room temperature for very long. I can definitely say that I face this problem almost every day when I'm preparing samples for any particular experiment that I want. Maybe protein purification using chromatography, maybe something involving mass spectrometry, something involving sequencing, or immunoprecipitation. You have to be extra careful with each of those. It's not possible to treat them at ultra cold temperatures, but as cold as you can keep them to prevent any kind of degradation. Use proper protease inhibitor cocktails or proper DNases, RNases, whichever step of the way that gets included or incorporated into your lysate. And be very careful while handling whole cell lysate. So there are steps, there are precautions, and I think there is a lot of reading material available that you can use and exercise your experiments with caution. So, yeah, these are the very basic things that we should keep in mind while preparing samples. So, yes, degradation definitely happens. Be careful.
You talked about how quickly the technology is improving.
Yes.
Uh, Specific to the work in malaria, I was curious if you could talk about how advances in chromatography mass spec could assist and improve the research in this area.
Yes. So anything related to biological sciences is related to therapy. So we are here to find a cure, we are here to treat something eventually, right? The idea now revolves more around specific targets. And by specific targets, we mean it could be a protein molecule, could be an enzyme, could be an RNA, could be a DNA. For example, when I talk about cancer, my main target is a long non coding RNA that I'm trying to characterize. In case of malaria, I'm trying to, for this particular lab that I'm working in, I'm trying to characterize kinases. So, identifying all these different domains in recognizing a particular target molecule, firstly, is very important. And when you're up against that particular molecule, to target it more specifically so that we can achieve the best of results in terms of alleviating the symptoms for that particular stage, it starts from a culture work, then getting all those in vitro assays developed, and eventually getting into in vivo work where we test them in mice as well. So, if you have come across that recent paper, that particular compound was tested in mice as well in stage specific activity assays, and we tried to find out whether it was really effective in alleviating symptoms for that particular stage. So, as I said, target identification is very important. And when we get to the different in vitro studies, culturing cells under proper conditions, getting the extracts, because malaria is very stage specific. So, getting the extracts from each stage and then harvesting them in a way so that we can try to have that particular protein, that particular target quantified and its activity evaluated. For that, we definitely need a measurable amount of that recombinantly expressed protein. So it basically starts from designing that particular construct in a suitable vector, transforming it, getting into the recombinant protein expression using IPTG induction, and then you break apart the cells, you get your protein of interest through multiple rounds in order to have its interactors evaluated and validated. Then you need to go through something like tandem affinity mass spectrometry or turbo ID generation to find out that yes, it has these many number of interactors. And at the same time, you could do immunoprecipitation experiments as well, where, as I said, you could have a certain tagged expression of a protein in a certain malarial line, culture line, and after getting that protein in hand, you can use antibodies specific for that protein in order to understand that, yes, it is that particular protein of interest against which I'm trying to design something or which I'm trying to target. Real drug discovery still remains certain in a way that we can get these validated through in vitro as well as in vivo assays. And eventually, that could, see the light of the day, these compounds, by getting them incorporated in different clinical trials. So you definitely have heard about these different kinase inhibitors like imatinib, and which have been used in different cancer therapies previously. So the idea basically comes from there, repurposing all those kinase inhibitors against different kinase targets, which can be identified in malaria. So my research mainly focuses on that, and I'm building up my research portfolio in this lab in that direction.
Obviously, malaria is a pressing issue globally. In terms of introducing your work to researchers in general, what else would you like them to know about the work you've been doing at UCF?
Yes. Malaria is still a very pressing issue because there are still over 600,000 deaths happening globally every year, and more than a million people are getting affected. And although it's more concentrated in sub Saharan Africa, you have more falciparum malaria in Africa, you have more vivax deaths, elsewhere. It could be Indonesia, it could be parts of India. I've even heard about malaria cases in the US. So that could be some kind of a contraction that might have occurred at airports or some community event. Malaria still remains a big burden. And one of the main factors adding to it is drug resistance. So, if you have heard about the strain called 3D7, which is a subpopulation of the NF54 isolate, that's another main strain or a parental strain that was identified initially in Netherlands. But 3D7 is associated with artemisinin resistance, and artemisinin is still one of the frontline antimalarials recommended by the World Health Organization for primary treatment. These days, artemisinin is administered to patients in combination with other, slow acting drugs. So one is a fast acting and the other is a slow acting, and this combination makes therapy more effective. But due to emerging drug resistance, it's becoming very difficult to tackle these strains. So, in order to identify what is basically the bottom line of this drug resistance plaguing the burden of malaria globally, there was one mutation that was identified in the K13, Elch13 propeller domain in one of the proteins in the falciparum strain associated with artemisinin drug resistance in 3D7. And from there, several targets have been identified with respect to the 3D7 or artemisinin resistant strain in malaria. So during my PhD work, I concentrated on a protein called falciparum 2, which is a cysteine protease. And cysteine proteases have been studied as drug targets across different domains, different diseases. So cysteine proteases, as the name suggests, they are a class of enzymes. They have a cysteine residue in their active site. And based on that, they can catalyze the cleavage of other their substrates basically, and falciparum 2 in this case directly acts on the host hemoglobin. So one of the most interesting factors that comes out and that regards it as a very promising drug target is its direct action on hemoglobin. And some in silico work that I have done in this light with hemoglobin in my previous PhD research work allows me to understand how that mechanism might be unfolding. And interestingly, I found that it is not only falciparum 2, but a host of other proteins forming like a 200 kilodalton mega complex that helps in the cleavage of hemoglobin and eventually helps in releasing amino acids by separating heme from globin. Those amino acids help the parasite, in its growth, in maintaining its osmotic stability, motility, and all of that. Heme, being toxic, is the parasite kind of bypasses it by forming hemozoin crystals, which are eventually eliminated from the parasite. When I came into malarial drug discovery, and I worked on this very latest publication of ours involving that repurposed CDK-like kinase inhibitor. One of the very interesting aspects of it was its direct activity on preventing hemozoin formation. So we did one assay, like one of our collaborators did an assay called the hemozoin formation assay, and that was seen that by administering this particular compound at different doses, hemozoin formation was actually going down, which means that the parasite is probably not able to bypass the toxicity of the heme by forming less hemozoin. So, these small details and these small results, they actually build up a very good story in light of how we can use these compounds to treat something as, promiscuous as malaria, because it might go apart in different directions by having, just developing some kind of mutation, some kind of single nucleotide polymorphism in a protein being attributed to its drug resistance. But at the same time, since they have state specific activities, identifying a particular target from a particular stage is also very important in understanding how robust targeting that protein of interest can be. So everything is interconnected in that way, but at the same time, it's interesting to understand how biomolecules can be in their game and how you can identify these targets for your own research work in light of therapy. So, yeah.
No, it's super interesting, and I really appreciate how you're able to explain such a complex topic. What advice would you give to researchers who are just starting in their field and face some of the same challenges like mastering complex sample preparation?
Yes. So, the basic idea of biological research is do hands on research until and unless you're handling cells yourself, until and unless you're centrifuging that lysate, you won't exactly be able to understand what you're handling. Coming from somebody like me, trust me, I was so skeptical about getting into research. I had no idea since I did not have a background in research before. But I was always interested about handling chemicals, handling samples, and working in a lab. So that has always intrigued me in some way. And that particular interest kind of got me build my portfolio in a way that yes, I can say that I can do research. Probably I can, but that does not come with failing at maybe 100 experiments every day. So it keeps happening. So I would say keep your motivation alive. Whatever you're doing is for a very, very noble purpose, very noble cause. You're trying, although you might feel yourself like a drop in a huge ocean, but that contribution that you're doing by identifying a target, by trying to work with that target so that you can achieve those specific aims for your project, for your hypothesis, is actually building a huge portfolio in the preventative medicine world, in the therapeutic world. So that's one thing to go ahead with the motivation. And sometimes, yes, you can have very, very long days in your lab where you might be doing just one experiment, but you might need to troubleshoot for another 4 hours in order to understand where you might be going wrong. But since biological samples, they are a bit tricky to handle. You're working with this very complex milieu, right, where you have things ranging from RNAs to DNAs to proteins to minerals, and everything is there. So you're handling that entire pool of things, pool of molecules. So one molecule might interfere with the other molecule, and your target protein of interest or your target molecule of interest might get degraded. Be ready for that, but at the same time exercise caution. Go through your protocols very well. Prepare as many things as you can do beforehand to get that particular result that you're trying to achieve as smoothly as you can. Again, at the same time, nothing is as smooth as it sounds. Handle your samples with care, and you will surely achieve what you're planning to. That could be like 50% of what you might have targeted before, but it still goes a long way. So do not lose that motivation and keep working.
No, I think that's great advice. Because, yeah, it's going to be intimidating to start and then not losing that
Yeah.
desire to keep going and being okay with failing and knowing that it's part of your ultimate success and what you're trying to uncover. It's critical. Because if you give up at any of those points along the way, you're not going to get there, right?
No, you're not going to get there. And you need to have a team. So it's difficult to work alone in a lab. So make sure you have a team or at least somebody, with whom you can troubleshoot well, and things definitely become easier. Biological research is a lot about teamwork. Try to handle as many different kinds of samples as you can, because when you're doing something with recombinantly expressed stuff, then that has a certain bandwidth of being handled. When you're working with clinical stuff, for example, you're handling blood plasma, then it has a different way to be worked upon. Clinical samples, they are not like you can have multiple rounds of recombinantly expressing your protein and working with it, even if you go wrong. But once there is one clinical sample that you might spill, that might not work, then it's gone. So, yes, you have to be careful. I have learned all of this a very hard way, but with the help of my team, with the help of my supervisors, with the help of my guides and mentors, I'm definitely here. Try to read up as much as you can, try to gather as much information as you can on the work that you're doing. Critically thinking your own faults, your own shortcomings is also very important in order to go ahead. So do not worry about how much you have done, but try to gauge every little effect that your work may have on a broader aspect, on a broader perspective, and can bring about a huge change eventually. So, yes, we might all be very zealous about getting that Nobel Prize eventually, but you get a Nobel Prize every step of the way as you get ahead in research. So once you get one experiment done, the satisfaction that it gives you, it's nothing short of getting a prize eventually. So be happy, be satisfied, but at the same time, keep questioning, keep thinking how much better we can do. I think these are some of the basic things we should keep in mind while being in clinical or biological research.
That's really solid advice. Subhoja, thanks for joining me. I really appreciate your willingness to share with the audience, and
Of
I'll
course.
certainly include some links where people can learn some more about the great work that's going on at UCF. So I really appreciate it.
I'm glad and thank you so much for this opportunity. I was really glad to share my research and hope people were able to understand it. And I was as comprehensive as I could be. Hope anybody who wants to get into research, anybody who wants to have that academic taste driven identity, good luck to all of you. Every step counts, every baby step counts. So, yeah, good luck.